So I'm a rice geneticist working in Aberdeen, and I want to tell you about one specific project, which is a BBSRC-funded project related to water sustainability on rice. It's a very substantial project, which is why there is a big cast list here. But it's a response to the call that BBSRC made on the effects of environmental change on the soil-water interface, implications for food production and water supply. This is a picture of our experiment last year in Mymensingh in Bangladesh. So the project title is a genetic dissection of traits required for sustainable water use in rice using genome-wide association studies. So that's a very long title. Very, very briefly, the project is about the fact that in Bangladesh, to maintain food food production, they have used quite extensively dry season irrigation using groundwater. So this is an electric pump that's taking groundwater, and that is unsustainable because that groundwater is not being replenished every year. So a number of organizations are promoting a technique called alternate wetting and drying, in which the farmer irrigates less. So alternate wetting and drying is being promoted as a water-saving technique in which the farmer puts a tube into his field and lets the tube there, and he can look down that tube. He can let the soil dry out a little bit until he can see no more water in the bottom of the tube, and then we'll irrigate again. So that has been recently being promoted, but there are some questions about that technique which we are hoping to try and at least start to come up with some answers. And that is, are cultivars and genes optimized for that type of production system? How does it promote improved yield? Because the general consensus is that yield is actually increased by this technique, although that is not always the case. Is it sustainable? And what does it do to grain arsenic and cadmium? Arsenic especially is a huge problem in Bangladesh. And we have reason to believe that if the soil becomes aerobic, arsenic uptake will be massively reduced. But it may have the opposite consequence for cadmium. So these are the people who helped write the proposal, including Andy Mahag, who's now in Belfast. But we've got geneticists, we've got soil scientists, we've got physiologists, we've got Lancaster, we've got the International Rice Research Institute, the team in Bangladesh Agricultural University, and we've got a couple of breeders in India who have also contributed to the discussions and the supply of seed. So that's just a map of the world showing where we're doing where the partners are coming from. As I say, alternative wetting and drying is being promoted. This is a picture of an experiment in India on alternative wetting and drying where at times the surface becomes dry compared to the normal flooded situation. And if anyone can read Bangla, that is a leaflet that's being produced for farmers to try and persuade them to use this technique. One of the major techniques we're going to use is genome sequencing as a method to do genome-wide association studies, and this is just to highlight that the grant proposal was written at the time that the Shanghai group, led by Bin Han, demonstrated in a beautiful paper how you can use genome sequencing to produce SNP markers for association mapping. So that is what we intend to do, something much like they did in their paper in 2010. What we're doing in rice is we're concentrating on a specific proportion of the rice population called Aus. So I want to just explain why we're doing that. This shows you the genetic diversity in rice. And this thing here in yellow is the Aus subgroup. They're particularly interesting for what we want to do. It should make the GWAS a lot easier because we overcome the problems associated with population structure. But I think this slide maybe shows the best is that this Aus population have their evolutionary origin or domestication origin in northeast India, which is where the geographic focus for the project is anyway. And the other interesting thing about Aus as a subgroup of rice is that they are currently the donors of most of the abiotic stress resistance in rice. So, for example, submergence tolerance, phosphorus uptake efficiency, salinity, drought and heat tolerance. Aus cultivars are being used in the breeding programs. So this is a very interesting at the physiological level, this group. So the expected outputs of the project is a collection of genetically characterized Aus cultivars for a genome-wide association analysis in Bangladesh and northeast India. insights into the sustainability of the AW technique, for example, are soil nutrients being depleted, physiological and biogeochemical understanding of changes in yield determining processes with AWD. I should say that you speak to different people and they've got different explanations of why AWD works. You ask a soil scientist, you get a completely different answer from a physiologist. So we should shed some light on that. Low sulfur adaptation to AWD and for improved grain quality should be available for plant breeding, and hopefully we will get to good candidate genes for some of these traits. If you want to know more, we've summarized all of that in a paper that's just come out in Energy and Food Security. That is there for you if you want to find out some more. This is some of the early work we did to decide what to put into this thing we're calling the Bengal Assam ouse population. So there are 300 cultivars. Just a small number of them are Czech varieties which are not from the ouse subgroup, but the rest of these are all from the ouse population, which should allow us to do good quality GWAS on that material. This is a picture of it just over a year ago being grown in the nursery prior to transplanting, and this is the team a year ago who transplanted it in Mymensingh in Bangladesh. This is actually the experiment of last year, where we have the alternate wetting and drying plots, 4 replicates on this side, and then 4 replicates of continuously flooded on this side. And this is a close-up of the alternate wetting and drying. You can see the soil at some stages appears to be dry. Actually, if you walk on it, it still feels wet. We have, to try and condense the experiment, we have a very simple 1 row of Czech variety, 1 row of test, 1 row of Czech, 1 row of test. And there are 300 test varieties in the population. This is Mohammed, postdoc from Lancaster, who spent 9 weeks at the experiment doing physiological and soil measurements. And this is one of the tubes being put into place in the field. We're using, from Lancaster, they're using these DGT probes. These are those in place in situ, which should give us some soil chemistry dynamics, and also rhizome samplers, which can give us instantaneous data on soil pore water. And I have to thank Rafiq Islam, who is formerly the head of soil science in Mymensingh, and his younger lecturer, Mohammed Simon, who is really the person responsible for making sure all of this is working. I'll skip that slide. Just shows you what the AWD cycle looks like in terms of water. So this is soil water depth. This line is 0. So this is the first cycle from when we started to measure when water had left the soil surface, and it went down and down and down until minus 15 centimeters, and then it was rewatered, and then down and down and down, and then rewatered when it's working properly on about an 8-day cycle. And we actually did 4 cycles last year. And I'm just going to show you some of the data, because this is really live data, a lot of this, and a lot of the really exciting stuff hasn't come in yet. So this shows you the soil water content using a delta T probe. 5 centimeters, you can see the soil moisture drops pretty substantially towards the end of the cycle. At 15 centimeters, you're getting some dropping towards the end of the cycle, but deeper down, you can clearly see that the water is still there, so still perfectly normal. Some pore water analysis. This is just showing you for 2 things that we have measured so far. This is pore water arsenic. So this is the AWD, and this is when the cycles start, and there's a very substantial drop in pore water arsenic, which seems to be maintained during several cycles. Whereas the continuously flooding, that dropping doesn't seem to occur. And it's matching very closely the situation for iron in the soil, which makes some sense. This is some physiology looking at leaf elongation rate. AWD here is in yellow, and we have a lower leaf elongation rate at the beginning of the experiment. The first cycle, we have some change, a higher elongation rate at the end. There's a subtle effect of AWD here, which is not entirely straightforward. This is leaf ABA concentration. This is in the middle of a cycle. This is at the end of a cycle. This is after rewatering on the second cycle. And then the same thing on the third cycle. Especially, I think you can see here that during the middle and end of this third cycle, there is higher leaf ABA content. And the statistics say treatment and the position in the cycle significantly affect leaf ABA. So there is a fairly subtle effect of this treatment on leaf ABA. This is probably the most exciting slide. We haven't, insofar as what farmers care about is yield. And in our experiment, just looking at the Czech variety, there is a 9.7% increase in yield under AWD. And there's a 16% increase in straw biomass, and actually harvest index is significantly reduced by AWD treatment. We have only just begun doing the arsenic measurements. Claire and Nicole are doing that in Aberdeen. This is the grain arsenic. There's only a 9% reduction in grain arsenic, much less than we were anticipating. So it'll be very interesting to see how the test varieties get on. And there's a 64% increase in grain cadmium. These cadmium levels, by the way, are not high, so this is nothing to be alarmed about. But if you're growing them in a soil which has high cadmium, this may be a problem. Yield in the population. This shows you a very simple mean yield under AWD versus mean yield in continuously flooded. And this is the one-to-one line. So you very quickly look at that and you think, yeah, the AWD treatment has actually increased yield. And in fact, across the whole set, there's a 33% increase in yield. And if you want to see that in a little bit more detail, this just shows you the means with some the standard errors, just so you can see this, get some idea of the level of variation that we're seeing in this experiment. So some of them seem to be behaving rather interestingly. What's interesting here is a plot of yield under the flooded conditions against their response. And what this is showing you is the ones that yield well under normal conditions do not respond as much. So the ones that are responding the most to AWD are the ones that do relatively poorly under normal conditions, which I think is telling us something important. Looking in more detail at the population and just picking out one small part of the population, I'm just going to focus on this one little clade here and just show you some more data on one clade of 9 genotypes. This is the one that responds the most, but this is all of its closely related genotypes. And if you look at the response of just that clade alone, you find what we're really interested in is a significant variety by treatment interaction. So we're optimistic that we will be able to pick up the genetics behind the response to AWD. The genome sequence data has only recently come in, done by TGAC. The first run was 160 cultivars with a mean of 4.4x coverage. Second run has a slightly better at 6.8x coverage. Susan Fairley in Aberdeen will be taking that data, aligning it to the reference, and giving us SNP data. Alex Douglas will take the SNP data and do the GWAS. And then Ahmed Manan will be doing some systems biology on that. And just one slide on that systems biology. He'll take the sequence information. He'll take information about transport processes and photosynthesis, and information about biomass production, and try and work out if specific lesions in specific genes are going to affect specific processes, particularly, for example, grain arsenic uptake, and using systems biology like that. We're now into the second year. The experiment is in Mymensingh again, but we're also going to do it in Faridpur and in Rajshahi. So very quickly, some photographs of those experiments. That's 3 weeks ago. Well, these are all taken just a few weeks ago. This is the sowing of the experiment in Madhpur and Rajshahi. Just a couple more slides. Because I'm doing this for the BBSRC, I feel important to do some public engagements. So I'm taking this talk about this project called Rice Water in Bangladesh to schools. This is my wife's school, in fact. And that's very rewarding to be doing that kind of thing. We're trying to replicate AWD in the controlled growth room. So this is the experiment of Munmi, who's sat at the back somewhere, and it looks like we are successfully reproducing in a rather simple system an AWD response. So it's very interesting. Last slide. Halfway through this 4-year project, what have we got to do yet? Actually, the list is huge. But we've got to do the SNP database. We've got the grain analysis to do, grain and shoot analysis for year 1. And we've got some quite a lot of statistics to improve the data. Year 2 to do. We're doing transcriptomics in year 2. We'll do the ionomics. This is the measuring the grain and leaf ion contents. We've got to do root phenotyping, and when all that's in, we'll do the GWAS, the systems biology, and then we've got to combine the soil biochemistry data to make a story to really work out what is happening below ground in terms of soil chemistry, and combine the plant physiology and the transcriptomics data to really work out what's happening at the level of plant physiology and gene expression. And that is it.